Good morning, students. So today we are going to discussing the 10th class biology of Telangana state. So important activity come diagram based questions. So these questions should come in examination level as 2 marks, 4 marks and 8 marks. So these are most important in examination level from which they may chance to ask the questions. So these are totally activity come diagram based questions. So now let us discuss these and chapter wise. That is first chapter the nutrition, the food supplying system. Now the first activity come diagram based question is number 1. So that is what is being tested in this experiment? What is the aim of this experiment? So for this question, the answer is the main aim of this experiment is to test the presence of starch in green leaves. To test the starch in green leaves. So what is being tested? Means starch is being tested in the green leaves, either present or absent in the green leaves. So next question, what are the materials required for this experiment? So the materials required for this experiment is, so the Bunsen burner, tripod stand, wide-mouthed beaker, test tube, methylated spirit, and green leaves, and asbestos gas. So these are the materials, and also a chemical is needed, that is iodine reagent. They are the materials required for this to test the presence of starch in the green leaves. Next, the third question, what are the changes that takes place in the leaf after boiling? After boiling leaf, when it is inside the test tube in the presence of methylated spirit, this spirit makes the leaf into a pale white color, pale white color, and also it removes the chlorophyll. So after boiling, what happens? Chlorophyll will be separated from the leaves. Next, what is the chemical used for the testing of presence of starch? Iodine reagent or iodine solution is used to test the presence of starch in the green leaves. How do you interpret the results at the end of this experiment? So when we poured on the green, when these leaves when take out from the test tube after boiling and keep in a petri dish, after keeping in a petri dish this leaf, and when we pour the iodine reagent, the chlorophyll parts turns into blue black color. So why they are turning into blue black color? Because of in that region starch is present. So this is a result when we pour the iodine reagent onto the leaves where the chlorophyll is there in that region, the leaf turns into blue black color. Why? Because there presence of starch, we can see that. So this is about answers regarding to this activity come diagram based question. So next to go to the second one. Who conducted this experiment for this first time? So when you see this diagram, so this diagram is related to the bell jar. So now what we can see, this picture is totally the bell jar, 4 bell jars, 4 situations, 1, 2, 3, 4. So here, who conducted this experiment for this first time? Means Joseph Priestley conducted this bell jar experiment. What happened to the burning candle and a mouse in a closed bell jar? So when we kept a burning candle and a mouse in a bell jar, so in a bell jar, in this a bell jar, there will be a burning candle is there. There will be a mouse, rat is there. So what happens here? Candle will be put off and rat will be die. So because of absence of oxygen. Next, what was the observations when a candle, a mouse, and a mint plant were placed inside the closed bell jar? When we kept to these three, there will be a candle is burning, and a rat is moving, and a plant is living, green leaves. So these three will be survive. So continuously, candle will be burned, rat will be move, rat will or mouse will be survive, and the plant will be survive. Why? Because here what's happened when we kept a plant regulate oxygen. This oxygen is utilized by the burning candle and the rat, whereas rat and candle liberate the carbon dioxide. This carbon dioxide taken up by the plant. In this way, what's happened? Photosynthesis and respiration both are occurred. Then what happens? There will be exchange of gases will be takes place. All these three will be survive or live in this when we kept these three. And what can be concluded from the above experiment? So the main conclusion is uh, due to when we kept a mint plant, it provide oxygen. Because of providing oxygen, what happens? Candle will be burned continuously, rat will be moved continuously live. So because of oxygen coming from the plants due to process of photosynthesis. Next, the third one, what is the aim of this experiment? And when we see this picture, so the question, what is the aim of this experiment? So the main aim of this experiment to conduct the CO2 is essential for the photosynthesis. CO2 is essential for photosynthesis. So what is the aim of this experiment means? How can we prove the CO2 is essential for the photosynthesis? What are the materials used in this experiment? There will be a healthy potted plant which is having long and narrow leaf. The leaf is must long and narrow leaf, and a healthy potted plant which is having long and narrow leaf, as well as a wide mouthed glass bottle and a wooden block, and there will be a solution. The chemical is called KOH. These are the materials we need. So third question, what is the chemical? What is the chemical used in this bottle? Why do we use it? So in this bottle, we use KOH, potassium hydroxide. This chemical we use to absorb the carbon dioxide. Whatever carbon dioxide which is present in the bottle for the absorption, we add the KOH. So KOH is the main chemical to absorb the carbon dioxide. What is the inference you drawn from the above experiment? So the main result or inference is CO2 is essential for the photosynthesis. Without the CO2, there is no photosynthesis. How are the operatives arranged? So first of all, let us take the healthy potted plant, which is having long and narrow leaf, and bring towards the plant, there will be a wide mouthed glass bottle, and inside the half of the leaf inside the bottle and rest of the leaf outside the bottle, and keep the rubber cork firmly, and also wooden block keep uh, below the bottle so not to fall down. After iodine test, which part of the leaf turns blue black color and why? So the the leaf of the half portion of the leaf which is present inside the bottle is turns into blue black color. The part of the leaf which is present outside of the bottle, which does not turns into, sorry. So here the part of the leaf which is inside it does not turns into blue black color. The out of the bottle, the part of the leaf which is present outside the bottle, so this part of the leaf is turns into blue black color. Why? Because it receives the CO2. In whatever leaf part which is present inside the bottle does not turns into blue black color. Why? Because it not received the carbon dioxide. So why do we need to discharge the plant? How to discharge the plant? So before conducting experiment, the potted plant must be kept in the dark, free from 2 to 3 days. Leaves are free from starch. So why do we Destarch means removal of starch. So whatever starch formed in the leaf before conducting experiment, we have to remove. To remove starch which is present in the leaf, we have to kept in the dark place. When we kept a dark place, whatever starch is present, totally completed. The empty leaves of the plant, we have to conduct the experiment. So by keeping in the dark room, in dark places, we should remove the starch. In this way, we can destarch the plant. Destarch means removal of starch from the leaves. How we remove means by keeping the plant in the dark room or dark places. So what is the now fourth one? The fourth activity come diagram based questions. Now the fourth one. So in which the first question is, what is the aim of the experiment? What is the aim of this experiment? The main aim of this experiment, how can we prove the oxygen is liberated during photosynthesis? How can we prove the oxygen is liberated during photosynthesis? What are the operators? How are the operators arranged and label the parts? So let us take the beaker and pour the water and take the hydroelectrics and insert into the thistle funnel and take the test tube, and the thistle funnel is inserted into the test tube and inside inverted and keep into this in this way in the beaker. So now the main label the parts is when you take, so this is the thistle funnel, hydroelectrics and test tube and beaker, water, and while we are conducting experiment, there is a gas bubbles are releasing from the hydroelectrics. These are the hydroelectrics bubbles. So how do we use only hydrilla plant, any aquatic plant for this experiment? So if you take instead of hydrilla plant, if you take another plant, we cannot prove this experiment. So why? Because of that, because of the ability to produce to conduct this experiment by using only hydroelectrics. So hydroelectrics by in this experiment only we have to use the hydroelectric plant twigs only. If you use instead of hydroelectrics, if you use another plant, we cannot to prove the result. That is, you have to keep in your mind. What precautions to be taken while doing this experiment? Main precautions: inserting of hydroelectrics into the thistle funnel and lifting of the thumbs up. Sorry, lifting up the test tube from the beaker. While lifting up the test tube from the beaker, you have to close the test tube mouth with your thumb and lift up and make right, and slowly you have to open. So to collect after sufficient collecting the gas bubbles. So lifting of test tube from the beaker, it is very careful. That is most important. And what is the gas accumulated in this test tube? Main gas accumulated in this so what is the gas accumulated in the test tube means the main gas is oxygen. Oxygen is accumulated in the form of bubbles. So how do you test the accumulated gas and interpret it? So carefully you have to lift up the test tube by closing its mouth with thumb, and near the mouth you have to ignite or light the matchstick and keep the near the mouth of the test tube, and the matchstick will glow vigorously and brightly. So how it is brightly and vigorously means due to oxygen only. Where do coming from the test tube? So where do we collect it? From the hydrilla twigs. So in this way, we can interpret, we can conclude the hydrolytic twigs liberated during photosynthesis, oxygen bubbles by igniting matchstick near the mouth of the test tube. It glows vigorously and brightly because of only in the presence of oxygen. This oxygen is liberated during photosynthesis. By this, we can conclude. And the next fifth one, what is the aim of this experiment? And main aim of this experiment is sunscreen or light screen experiment. Light screen or sunscreen experiment. Why do you close a pot of leaf with black paper and light screen? So we must take a designed the black paper or light screen because of not to receive the light to the closed pot. Only we are testing light focusing or light receiving part and light not receiving part. So to know the main important, this is either during photosynthesis light is essential or not. So this experiment, the main aim of this experiment is to how can we prove light is essential for the photosynthesis. So we have to close the leaf by taking black paper. Why? Because whatever we cover the black paper and it does not receive the sunlight, whereas the cut design, a spot inside a spot only receives the sunlight. Now testing this, how do you interpret the result of this segment? So after covering the black paper onto the leaf with taking cut design black paper, and after remove the black paper and test both the designed part and covering part. The covering part, this is the covering part, does not turns into blue color. Why? Because it not receive the light. The cut designed S-shaped part will receive the light. That is the reason this part turns into blue black color. So the part which is open that receive the sunlight and prepared the starch. The part not received the light, it not turns into blue color. Why? Because not prepared the starch. In this way, we can conclude that light is essential for the photosynthesis. How do you test the presence of starch in the leaf? By taking iodine reagent, adding both parts, the covered part and opened part. The covered part does not turns into blue color, and whereas the open part is turns into blue color. This indicates that light is essential for the photosynthesis. So now the sixth one, name the diagram and label the parts. So the diagram is one of the important cell organelle which is present in the plant cell, the chloroplast. This was discovered by Skimper scientist, Skimper. So chloroplast discovered by the Skimper scientist. So this cell organelle is most important to carry out photosynthesis. The main, it is oval shaped and surrounded by the double membrane, oval shaped double membrane, and it has thylakoid, and liquid part is called stroma. This one totally stroma, and coins like structure which are arranged one by one. These coins like structure arranged one by one are called thylakoids, as these are arranged in a grana. So hence these are called grana thylakoids. So now these are the grana thylakoids. These one group of grana thylakoids and another group of thylakoids attached with the help of stroma thylakoid. Stroma thylakoid. On the grana thylakoid membrane, photosynthesis pigments are present. Reaction centers are present. In the reaction centers, because of presence of chlorophyll pigments, in that region, the photosynthesis will be carried out. So where are these cell organelles found? These organelles are found in mainly greener parts of the leaves, mainly greener parts of the leaves. And what is the main function of this structure? So the chloroplast the function is the main important function to carry out photosynthesis. and also it convert to the light energy into chemical energy, and also it participates in photolysis of water, and it turns into carbon dioxide into glucose. Carbon dioxide into glucose. These are the three main functions regarding to the chloroplast. And seventh one, what does this diagram indicate? Uh, what is the shapeless organism drawn in this diagram, and is it nutrition or excretion, and explain the intake of food by the organism. So the main, so what does this diagram indicate? So this diagram is indicate that amoeba takes the food by using temporary finger-like exchanges like pseudopodia. So the mainly, what is the shapeless organism drawn in this diagram means amoeba. Here the question answer is amoeba. So here the digestion one, and inside there will be a food vacuole. Complex substances are broken down to simple ones, then they diffuse into the cytoplasm. So when they take this food material, they may diffuse into their bodies, and into the cytoplasm. The whatever undigested material will be remain moved onto the surface of the cell and thrown outside. So with the help of pseudopodia, they are absorbing the food material. So the total process, so this belongs to nutrition only. Is it nutrition or excretion? Means nutrition only. Explain the intake of food by the organism. By the process of diffusion, with the help of pseudopodias, they may take the food materials. Next, eighth one, name the diagram and label the parts. So the main important of this diagram is human digestive system. And when you take the mouth, esophagus, mouth, pharynx, and esophagus, stomach, liver, pancreas, duodenum, small intestine, large intestine, here they given, large intestine, small intestine, rectum, anus. Rectum, anus. What is the main functions of this organ system? The main organ system is digestion. So food materials are broken down into macromolecules into micromolecules by the human digestive system. Particularly, a organ is called food pipe or gut or alimentary canal. Explain the life process that takes place in this organ. So the main life process is digestive system. Draw the flow chart showing the different parts of this diagram and the changes that takes place. So first of all, let us take the mouth, and then oral cavity or buccal cavity, and then then pharynx, and then esophagus, and then stomach, and then small intestine, large intestine, and rectum, anus. This is the sequence of food processing from mouth to anus. And what are the changes? Mainly physical, chemical, both changes that takes place. Physical changes with the help of teeth and wall muscles of the digestive system and gut. Chemical changes by the enzymes are occurred. So now, what does this diagram indicate? Name the part of alimentary canal in the diagram. Which muscles are responsible for such movements? So this diagram is indicate that there will be a peristaltic movements. Peristaltic movements. This part is esophagus. So in the movements, wavy manner movements of football is called peristaltic movements, and muscles muscles are particularly so circular muscles, vertical longitudinal muscles which are present in the esophagus responsible to movement of the football in the esophagus. Next, 10th one. What are the two glands present in this diagram? So one gland is the liver, another one is the pancreas. What are the juices secreted by the two glands? So liver produce bile juice, liver produce bile juice, and pancreas produce pancreatic juice. Pancreatic juice. Liver is right side, pancreas is left side, and the liver produces bile juice. It does not have any enzymes, whereas pancreatic produce pancreatic juice and have the enzymes. What are the contents of enzymes present in the juices secreted by the both glands? So in bile juice, in there is no enzymes. Only pigments are present, bilirubin and biliverdin, which gives yellow color. Whereas pancreatic juice consists amylase, trypsin, chymotrypsin, and these are breakdown into fats, carbohydrates, proteins. So amylase, pepsin, trypsin, chymotrypsin, and lipases enzymes are present in the pancreatic juice, which help to digest the fats, carbohydrates, and fat proteins. So with this, there will be a first lesson, nutrition. Important to diagram come activity based questions answers are completed. So you once you go through that, take a textbook and practice well, and your own way you can write down, take a notebook and write down these questions, and as well as while preparing the textbook answers also you should write. It is most important in examination level.